This last segment of the course is all going to have to do with material transport, movement of different materials, both into the body from the environment and from the body back out to the environment in the form of wastes. So when we're thinking about this idea of material transport, we need to keep in mind that we're transferring resources from the environment, but we're transferring it actually to each and every one of the cells in the body. When we eat, we're not feeding our stomach. When we breathe, either by gills or by lungs, we're not getting oxygen just to those organs, but remember, these need to be distributed to every single cell in the body that needs these materials to survive. And so similarly, when they undergo their cellular respiration and produce metabolic wastes, we need to move those from the cells back to the environment. So we always have to remember that this is all happening kind of at the macro level, but the end result is down here at the cellular level. So the way these materials get into and out of the cells is by diffusion. But remember back to when we were talking about allometry and scaling, as body sizes get larger, we have a decrease in the surface area to volume ratio. And so whereas very, very small animals can do this material exchange directly by diffusion with the environment, what we see in large vertebrates is this combination of bulk transport, bulk flow, to move these materials throughout the system, and then diffusion once you get down to the level of the cells to get that material into and out of the cell. So in this web lecture, we're going to focus on the diffusion side of things, and we're going to use Fick's law of diffusion, which is a mathematical description of the rate of diffusion, as a framework to understand the mechanisms for maximizing the material transport in these multicellular, large-bodied vertebrates. So let's first just take a look at the different variables in Fick's law of diffusion. Again, this is describing the rate of diffusion. The capital R refers to that. The capital D is a diffusion coefficient. It's going to have to do with the medium that diffusion is occurring in, the temperature, different aspects. We're going to treat this more or less as a constant with respect to the different adaptations of vertebrates, so we're not going to talk very much about that diffusion coefficient. What we are going to concentrate on are the different adaptations for manipulating the rest of these factors in the equation. So capital A is the surface area available for diffusion. This is going to be a big one when we're talking about providing nutrients and oxygen to a very large-bodied animal. You need to really get a lot of surface area. Delta P is the difference in partial pressure of the diffusing gas. We can also think of this as just the concentration of the material that's being exchanged between the two sides of the surface or wherever that diffusion is occurring. What's the difference in concentration, or if we're talking about a gas, the gas partial pressure. And then finally, in the denominator, we have diffusion distance, so that we can see that there's an inverse relationship between the rate of diffusion and the diffusion distance, such that the greater the diffusion distance, the lower the rate, the lower the diffusion distance, the greater the rate. So let's look at these three factors in turn and think about the different structures and adaptations that we see in vertebrates for optimizing each of these different components. We're going to start with the surface area for diffusion. And let's first think about gills. So in this figure, what you're seeing on the left-hand side is a very early developmental stage in a shark of the development of these pharyngeal slits that are common, at least in some developmental stage, in all vertebrates. And then on the right-hand side, we see the definitive adult structures in place. So what you're seeing is a coronal section or frontal section straight through the oral cavity, going into the esophagus, looking as if the head of the shark is separated from front to back. So what we're seeing here in the early stages of development is that these gill slits are going to develop both as an in-pocketing from the inside of the pharynx and also in an invagination from the body surface. So the blue, remember, is the ectoderm, the yellow is endoderm, and they're going to sort of grow inward toward each other until these in-pouchings meet and form a complete slit from the pharynx all the way out to the external environment. Remember that in gnathostomes, the first two arches numerically become the jaw for the first one and the hyoid for the second. So the actual first gill pouch is associated with the third visceral arch. So this is what you're seeing here in cross-section in this view. And then what we're seeing next to them are these blood supplies, an individual one for each compartment, and we call these aortic arches. So each of these aortic arches are seen here right next to each pharyngeal arch. And these are bringing blood from the heart to the gills to become oxygenated. Each of these little interbranchial septa, these flaps of tissue in between these pharyngeal slits, is supported by a gill ray. So a little skeletal element that is supporting this interbranchial septum. Protecting these little gill chambers are these little flaps of tissue called gill rakers. And so what these gill rakers do is they prevent any sort of larger items such as bits of food or anything that might get caught in these fine gill filaments from entering the gill chamber. So this is sort of a filtering mechanism. And then attached to each side of these interbranchial septa are the actual gill lamellae. So this is the tissue that actually makes up the gills. And so we can see this in a photograph of a fish with its operculum being lifted. Remember, the operculum is this bony flap that covers the gill chamber in all osteichthyes. And so what you're seeing here is the branchial arch supporting the whole gill segment. You can see the gill rakers sieving out larger pieces of things to keep them out of the gills. And then finally the gill lamellae, the gill tissue itself, each of these supported by a gill ray. So let's take a look at the structure of these gills diagrammatically. First of all, let's just point out the branchial arch. So this is the skeletal structure. We're seeing it in this view in cross-section, one for each interbranchial septum. And then these flaps of tissue are called the primary gill lamellae. So this is the first level of elaboration of that surface area, these flaps of tissue that are going to increase the surface area of these gills initially. And each one of these primary gill lamellae bear additional elaborations of the surface, these additional flaps of tissue on each primary gill lamellae called secondary gill lamellae. These secondary gill lamellae are where the actual diffusion is going to occur. This is where the gas exchange is going to occur on these little flaps of tissue. So you can see with these different levels of elaboration of surface area. In fish gills, we get a very, very huge overall increase in surface area for gas exchange, maximizing that area term in Fick's equation. So now let's think about gas exchange in air and how lungs will increase the surface area. So remember that the concentration of oxygen in air is much, much higher than it ever is in water. And so air-breathing lungs do not necessarily have to be as efficient as gills are. And we'll talk a lot about different ways in which the efficiency of oxygen uptake in gills is maximized. So in amphibians, what we see are the lungs are basically just big air sacs with kind of a convoluted surface that increases the surface area of that sac. So relatively low surface area, but an increase over what you would see in something like a lungfish, which is just kind of a bag full of air, highly vascularized. In the sauropsids, we see something called a faviolar lung. So if we start from this starting point with just little folds in the surface of the sac, what we start to see is more and more subdivision, more and more infolding in these faviolar lungs. These increase the surface area by just successive levels of invagination of the surface. This increases all the way up to birds that have a highly elaborated faviolar lung with tiny, tiny little air capillaries, networks of little air capillaries that are the result of more and more elaborate folding of this simple sac-like structure. What we see in the synapsid lineage is something called an alveolar lung. So the alveolar lung is based on sequential branching of tubes. So we see more and more little branches of this initial tube structure ending in these little tiny bubbles of tissue at the very ends called the alveoli. So we can see that better in a diagram of human lungs. And we start with the trachea, which is going to branch into a pair of primary bronchi, one to each lung. Each of those is going to subdivide further into secondary bronchi and branch and branch and branch. And so you can think of this development as just kind of like the way a tree grows, with more and more branching until you get out to these very fine bronchial tubes that are going to end in these little bubbles of tissue that are going to be surrounded by blood capillaries, and this is where the actual gas exchange is going to occur. So again, elaboration of the surface by this branching pattern leading to millions and millions of these tiny, tiny, tiny little air bubbles, which are the sites of gas exchange. So now that we've talked about the surface area for diffusion, how this is maximized, let's look at how the diffusion distance is minimized in vertebrate gills and lungs. So let's start with the gills. So if we look at the structure of one of these secondary lamellae, so these little flaps of tissue, basically we've got a tissue that's just a couple of cell layers thick with these blood vessels sandwiched in between them. So this brings these tiny little capillaries, and these capillaries are so narrow that basically red blood cells go single file through these tiny little capillaries. And so each red blood cell is in very, very close proximity to this water that's flowing in between these gill lamellae. So this is how the diffusion distance is minimized. All it has to go through is just a couple of cell layers to exchange material with that blood. Similar situation in the alveolar lungs of mammals. You get these networks of capillaries, again, so narrow that these red blood cells are going to go through them single file, very close proximity, and they're going to be in these tight mesh networks surrounding each and every alveolus, each one of these bubbles. And then these alveoli, again, are just one or two cell layers thick, a very small distance to go between the air inside and these blood vessels around the outside. Finally, let's think about how this difference in partial pressure of the diffusing gas between the two sides of the surface can be maximized. And we're going to think primarily about water breathing with gills, because really with air breathing, the difference in partial pressure is always huge. There's always a lot of oxygen in the air, and there's always going to be sort of this maximum rate of diffusion between the deoxygenated blood and this very oxygen-rich air in the lungs. So we're just going to think about breathing in water. So let's look again at the structure of fish gills in diagrammatic form. So we've got the deoxygenated blood coming from the heart, and it's going to be delivering this blood to these secondary lamellae. So this gray tissue here is the primary gill lamella. This is the interbranchial septum, and the water is going to be coming through the septal channel. So the water is coming through in this direction, through the septal channel, in through the mouth and out through the pharyngeal slits. The blood is being brought through in the opposite direction. So here's the deoxygenated blood coming, breaking down obviously into finer and finer blood vessels until we get to these very small capillaries, and it's flowing in the opposite direction from the oxygenated water coming through. So this sets up a system called countercurrent exchange. So when these fluids are flowing in opposite directions, the best way to really get your mind around countercurrent exchange is to first think about what happens if we didn't have this arrangement, if these fluids were flowing concurrently or in the same direction. So what we see here is the water is coming in 100% oxygenated. The blood is coming in, let's say, completely deoxygenated, just for the sake of this illustration, and they're flowing along together in the same direction. So what we have here is a very, very a high difference in partial pressure between the oxygenated water and the deoxygenated blood. There's going to be a very high rate of diffusion initially. But as we go along this area of contact, the concentration is going to become more and more similar due to this high rate of diffusion, and you're going to get a little bit less exchange, until eventually the blood and the water reach an equilibrium. They're going to be at equal concentrations, and you're going to get basically about 50% saturation of the blood with oxygen. So it'll reach an equilibrium. Once it gets to equal concentrations in the blood and in the water, oxygen molecules will still be moving, but they're just as likely to move from the blood into the water as they are from the water into the blood. And so there's going to be an equilibrium with equal rates of diffusion going in either direction, and this blood oxygen saturation is going to remain around 50%. With countercurrent flow, now if we imagine the water flowing in this direction, the blood flowing in that direction, the water comes in at 100% saturation. The blood is coming in from the other direction at 0% saturation. If we think first where the blood is coming in, the water is pretty nearly depleted of oxygen from having been in contact with the blood all this distance, but it's not quite zero. It's not going to be quite down to the level. So at this point, oxygen is going to flow from the water into the blood at a low rate. There's just a very small difference in that partial pressure. But there's going to be this difference in partial pressure all the way along. So if we think of where the water is coming right in, the blood is going to be nearly saturated but not quite saturated. It's still going to be a little lower concentration of oxygen than we're finding in the water. So there's still going to be oxygen flowing from the water into the blood all the way along this entire contact distance. There's going to be a flow of oxygen into the blood. And so instead of getting just about a 50% saturation in the blood, as in the case with concurrent flow, with countercurrent flow we get near maximal saturation of the blood with oxygen. And so looking at this in graphical form, we see the situation with concurrent flow. There's going to be a huge amount of diffusion from the water into the blood at first, due to that large difference in partial pressure, but eventually there's going to be this equilibrium reached with a relatively low saturation of oxygen in the blood. This is the situation with countercurrent. At every point along this distance, along the gill lamella, there's going to be some difference in partial pressure. The oxygen is going to keep flowing from the water into the blood, and we can see that we get a very much higher ultimate concentration of oxygen in the blood. So remember, with these transport systems, it's really the total amount of oxygen we're getting loaded into this blood that matters, as opposed to a situation where you've got diffusion going into a cell and the rate of diffusion has to match the rate of metabolism within that cell. In that case, the rate's going to matter a lot more. And you'd want this kind of situation with a high rate of diffusion. But if your goal is to just get as much oxygen loaded into that blood as possible, it doesn't matter that there's just a small rate at every point along the way. What matters is that you get this really high level of loading of oxygen into the blood. So this is your introduction to Fick's law of diffusion, all of these factors that are going to contribute to effective diffusion between two surfaces. We've seen the ways that the surface area for diffusion is maximized by these different elaborations of the gills and the lungs. We've seen that when you come to an actual diffusion surface, such as the secondary gill lamellae or these little alveoli in the alveolar lung, we've got a very, very small diffusion distance. So these molecules of oxygen don't have very far to go to be able to get from this air or water into the blood. And we've seen that fish can play around with manipulating this difference in partial pressure to maximize the amount of oxygen that's loaded into the blood by using this countercurrent exchange system.